back to Devans Biology. In this video series on respiration, we have already cleared the concept of respiration as well as we have discussed or we had a brief look on the mechanism of respiration. Today in this video, we will discuss in detail the process of glycolysis, that is the first stage in the mechanism of respiration. Glycolysis is actually formed by two important words, glycon and lysis. Glycon means sugar and lysis is splitting. So, in short, it's splitting of sugar. Glycolysis is a splitting of sugar. Glycolysis is actually also known as EMP pathway. EMP named after their discoverers. EMP. Emden, Meyerhoff and Parnas. These were the three discoverers of this pathway. So this is also known as EMP pathway. EMP pathway is actually the most common pathway. The other pathways are ED pathway and HMP. ED, again this is named after their discoverers Entner and Duodroff. And the other pathway is HMP. HMP means hexose monophosphate pathway. Now the glycolysis is actually a partial oxidation of glucose to pyruvic acid. This is performed, glycolysis is performed in the cytoplasm of the cell, or we can say cytosol of the cell. What is a cytosol? It's a cytoplasm without the organelles. So glycolysis occurs in cytosol of the cell. All the living organisms undergo glycolysis, either they are aerobic or they are anaerobic. But the glycolysis is the only pathway that is being followed by anaerobic organisms, because after glycolysis the next stages needs presence of oxygen. There are actually main 10 steps that occur in the process of glycolysis. The first half of the glycolysis process is glucose activation phase, and the second half is payoff phase, that is energy producing phase. In the first step, glucose is converted into glucose, which is a 6-carbon compound, is converted into glucose 6-phosphate. Means glucose is phosphorylated to glucose 6-phosphate. Which is the enzyme used? The enzyme used here is hexokinase. The enzyme used is hexokinase. Now this kinase enzymes actually adds phosphate to the compound. As this is getting phosphorylated, so ATP will get converted into ADP. This hexokinase enzyme is actually inhibited by the excess of glucose 6-phosphate. This causes the cell to store glucose. Now the next step is glucose 6-phosphate gets isomerized to fructose 6-phosphate. As isomerization is going on, so the enzyme used is phosphohexoisomerase. The next step is fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate. Right. Now here the enzyme that is being used. The enzyme used is phosphofructokinase, also known as PFK. Now this enzyme is also known as pacemaker of glycolysis. You know what is a pacemaker? Pacemaker regulates the heartbeat. It means it can increase or decrease the heartbeat according to the requirement. Similarly, this enzyme is used as a pacemaker of glycolysis, which controls the step of glycolysis. In glycolysis, most of these stages are reversible. This step is also known as rate limiting step. Rate limiting step is usually slowest step of the whole process. This step is inhibited by the excess of ATP. Also, this step is the slowest step in the whole process of glycolysis. Now next, fructose gets split up into two different compounds that are glyceraldehyde 3-phosphate, also known as phosphoglyceraldehyde PGAL, and DHAP dihydroxyacetone phosphate. These two are interconvertible. This reaction is done or catalyzed by the help of enzyme aldolase. The aldolase enzyme splits the aldol into two different molecules. Now, as I told you, these two are interconvertible. So, the enzyme used here is phosphotriose isomerase. Right. Now, in the next step, these steps are glucose activation steps, or this is the phase of glucose activation phase. Right. From now onwards, there will be energy producing phase. Here, one molecule of glucose gets converted into one molecule of glucose 6-phosphate, one molecule of fructose 6-phosphate. It is converted into one molecule of fructose 1,6-bisphosphate. These all are 6 carbon compounds. Now this is a 3 carbon compound. That is why it's a triose isomerase. This is a 3 carbon compound. This is also a 3 carbon compound. So this 6 carbon compound will form 2 molecules of 3 carbon compound. Right. Now next step is will get converted into 1,3-bis phosphoglyceric acid. The enzyme that catalyzes this reaction is diphosphoglyceraldehyde dehydrogenase. In this reaction, NAD is reduced to NADH plus H plus. The dehydrogenase enzyme, what is the function of dehydrogenase enzyme? It removes hydrogen atoms instead of oxygen in oxidoreduction reactions. You must know the functions of all the enzymes that are being used in the mechanism, right? Now the next is, so this is also 2 molecules of 1,3-bisphosphoglyceric acid, right? Now this 1,3-bisphosphoglyceric acid will be catalyzed by the enzyme phosphoglycerate phosphorylase, as this is enzyme, so it will end in ASE, right? Now what this transphosphorylase enzyme do? Transphosphorylase enzyme catalyzes the transfer of phosphate group from one molecule to another, right? Now here ADP converts into ATP, and what is formed? The compound that is formed is 3-phosphoglyceric acid. You are supposed to note down all the functions of the enzymes that are being talked here, right? Now the next step, this step, one more important thing, this step is a substrate level phosphorylation. What do you mean by substrate level phosphorylation? It means direct synthesis of ATP from metabolites. Right? This is the point to be noted, that this step is substrate level phosphorylation. Now, in the next step, 3-phosphoglyceric acid converts into 2-phosphoglyceric acid or 2-phosphoglycerate. The enzyme that catalyzes this reaction is phosphoglyceromutase. Again, I forgot, all the reactions are reversible reactions except this first one, third one, and the last one. Mutase is an enzyme of isomerase enzyme which catalyzes the movement of phosphate group or movement of any functional group from one place to another in the same molecule. Here the phosphate group was at the third position, here the phosphate group is at the second position. Right? Now, next 2-phosphoglycerate converts into phosphoenol pyruvate. This reaction is catalyzed by enolase enzyme. I am writing the enzymes in red. Enolase enzyme. Now, what is the function of enolase enzyme? This enolase enzyme manages the reversible reaction between 2-phosphoglycerate and phosphoenol pyruvate in glycolysis and gluconeogenesis. Now, the last step is conversion of PEP to pyruvate or pyruvic acid. Right? This reaction is catalyzed by pyruvate kinase. Pyruvate kinase. In this, ADP gets converted into ATP. This is also a substrate level phosphorylation. I have already discussed what is the substrate level phosphorylation in this step. Right? This step is not reversible. So the steps which are not reversible, they are actually rate limiting steps. This enzyme is inhibited by the excess of ATP or high levels of ATP. So these are the total 10 steps in which the first half was glucose activation phase and the second half is energy payoff phase or energy releasing phase. In this reaction, in this process, then total 2 ATPs are being utilized. 2 ATPs used. How many are formed? We know that here 2 molecules were formed. So, here the ATPs formed will also be 2 in number. So, 2 ATPs here and 2 ATPs here. Total 4 ATPs are formed. 4 ATPs formed. I have tried to clarify the whole process. Still, if you have some doubts, you can write in the comment box. Your doubts will be clarified. Thank you.